Hi all, welcome to the NPTEL online certification program and welcome to the Biophotonics course. I will introduce myself. I am Jijit. I am an interdisciplinary research scholar of IIT Madras. My broad area of interest is protein biophysics. Here I am using the translocation and degradation of knotted proteins using single molecule flow spectroscopic techniques, mainly optical research. I am a student of chemistry and biotechnology department. So we'll start our session. We'll wait for some others to join for some time. Then we'll start. we'll start our session. Actually, this session we can do in such a manner that this will be a revision. So we'll go through the proper assignment which we people already solved. We'll start from discussing the questions and we'll go through the different manner, and during that time we can have some discussions. Okay, so we can see. So we'll start from the assignment zero. Okay, so these are the assignment zero questions. We'll discuss these questions and try to solve ourselves, and we will understand more things from here. Okay. Okay. Have a consideration also. Let's start with the first question. So these are like the basics items which may be required for that course. Yeah, people already had that idea. So we'll go through these questions and answer, and we'll discuss the some aspects of these things. So this actually, yeah, this thing actually we discussed once. So we will go through this one in a fast manner. A transistor is a dash control device. The question is, a transistor is a dash control device. It's a voltage control device, B current control device, C both A and B, D none of these. Answer: transistor is a current operated device. Since the, yeah, solution they given, that's a transistor is a current operated device, since the output voltage of the power is controlled by the input current. You can see how it is happens. So how it is actually getting controlled, how we can say that transistor is a current control device. See, transistor is a, transistor, or you call bipolar junction transistor. Transistor has three terminals. It's called emitter, base, collector. So these are the circuit representation, and this is a block diagram. Okay. So what happens when a transistor is connected in a circuit? So, so let it be a P transistor or an NPN transistor. So what happens? So the transistor equation is written IE is equal to IB plus IC. So this is actually coming from our Kirchhoff's current rule. So current IC will come from here, IB will come from here, and that will actually given here as IE. So the emitter current is a sum of collector current and the base current. This is the typical transistor equation. But the thing is that the current IB, the current flow through this base is actually helping to flow the current from collector to emitter. So this is actually when the transistor is biased according to a requirement, actually the current flow through the base is actually controlling the current flow from collector to emitter. So and the current which we can convert to voltage using a resistor, because we will get a potential drop across the resistor when some current is passing through that one. So according to that, we can manipulate our voltages connect to the transistor. So, but typically the transistor is a current device. The thing is that whatever is getting control is actually current, and the voltage or the current flow through the transistor is also controlled by the current which is passes through the base. So typically we can see a transistor is a current control device. So there are voltage control device which is called a field effect transistor. A common field effect transistor will have a, so what happens in the case of field effect transistor? There are a channel like a source drain channel will be there, and there will be a gate. The gate is actually the source drain channel will be like a common channel through which the current can pass. But the gate will have an opposite potential. So when we are biasing this one, what happens? This gate will actually grow inside the source drain channel, and actually it will, there will not be any current from this gate to the source drain channel. Always this is a growth actually hinders the flow of electron from through this channel. So and how this region is growing, that is actually controlled by potential. Since there is no current flow, the potential difference across this gate and source drain channel is getting increased, and that's why it is the getting controlled. So actually what is controlling the current flow? The current flow is controlled by potential difference. So the FET or field effect transistor can be considered as a voltage control device. Current flow is controlled by voltage. Okay, so transistor is a current control device. Field effect transistor is a voltage control device. Okay. Now going to the second question. Under normal condition, a diode conducts current when it is a forward biased, reverse biased, saturated, conducts in all cases. That also we already discussed here. So this will go a bit faster. So a diode will have a junction. So that is called junction diode. It's P-N. This P side is topped with the P-type semiconductor, and N side is actually made up of N-type semiconductor. So actually how it is made, there will be a single silicon chip on one side of that thing is actually doped with the P-type, and that side is actually doped with N-type. So that the diode is made. And the circuit symbol of the diode will be like this. Okay. When the P side is connected to positive side of the battery, the N side is negative side of the battery, I put a resistance, otherwise it will be short. So here also what happens is current can flow through this one. So current will flow through this way, and there will be a voltage drop across this resistor or something like that. Okay. So This kind of configuration where the p side is connected with the positive terminus of the battery and n terminus is connected with the negative terminus of the battery, then that particular configuration is called forward biasing of the diode. On the circuit side it will be like this way. So actually this symbol is represented by the flow of the current. So it will flow like this manner and it will act as a valve. It will not allow any current from this side. That means if it is a positive, it will flow the current in this direction only. And if it is negative, it will not allow any current in this direction. So the case of a normal diode, the current flow is actually happens when the diode is at a forward bias. So why it is saying like it is a normal circumstances. So there are different diodes, which I think you already know. That is one of the diode is Zener diode, which is represented by this. So the Zener diode is actually operates in a reverse bias mode. Why? Because usually in the diode, if you put a characteristics, you can see that as the this is current, this is voltage, and this voltage and current graph of a diode is called as a characteristics graph of the diode. In the case of a diode, what happens as the voltage is increases, there will be slight increase in this current. Then after a particular voltage, it's called knee voltage, current from diode will increase in an exponential manner. And this is actually the operating condition. So there will be high current will go through this diode, and if the diode has the capability to withstand that much current, then the diode will be in the operational condition in that region. And this voltage is called knee voltage. And above this knee voltage, the diode can act in a normal manner. But what happens if you put the reverse bias mode? So reverse bias mode, what happens? There will be slight increase because this minority charging areas and other things. But what happens after a particular voltage, this current will suddenly increase, and this is called the breakdown voltage. And after this one, the current flow in the current diode is so fast such that it will not use, we cannot use it as a diode. But actually, Zener diodes are specially designed diode which will always work in the breakdown region. Why it is important? You can see that in the case of forward biasing, as the voltage is increasing, current is also increasing, but it has a some slope. So there you can see it's a finite resistance. Some forward resistance will be uh, forward resistance associated with diode. But in the case of reverse bias Zener diode or reverse bias diode, the voltage is somewhat fixed. The breakdown voltage is somewhat fixed at a very high range of the current. So the slope is almost like infinity. So the so if it is working in this region, the voltage across the diode is always constant. So Zener diode can be used as a this voltage regulator. Okay, so that can be used as a voltage regulator and the breakdown region. So this is like that's why we are called under normal circumstances the diode will conduct electricity when it is in forward biased. But there are diodes which is working in the reverse bias mode also. That are the that is not the typical diode. Okay, so here the answer is forward biased. A semiconductor is formed by dash bond, anic bond, electrovalent bond, covalent bond, and none of this. So that also we already know that. Just think about what are the mainly semiconductor materials. So the main answer is the germanium and silicon is the thing. And which group it will come? It will come in 14th group carbon. What are the elements? Carbon, silicon, germanium, tin, lead. Carbon, silicon, germanium, tin, lead are the elements in that group. And these are, yeah, that group has a change. This carbon is in an inherent manner. This is like a insulator, but yeah, graphite is conductive. One of the amorphous of the allotrope of the carbon. Graphite is conductive. Silicon and germanium are semiconductive, and tin and lead are metals. So these are the main thing which we use as semiconductive. What's the valency of these things? So the valency of that one carbon is two. The configuration should be 2 4 or like 2 8 4, something like that. So the outermost shell contains only 4 electrons. So how it can arrange? So this all, so these atoms will arrange in such a way that so each atom will share 4 electrons with the nearby atoms. So octet configuration will be obtained. So this is the reason. So it cannot give all 4 electrons to one species and cannot take all 4 electrons from a species also. What it will do, it will share electrons. It will share all the 4 electrons of the outermost shell to the nearby atoms such that it will be in a covalent bond. When we are adding impurities called donors or acceptor molecules, that can create some extra electrons or extra holes like that. Okay. So the answer of this question is it's a covalent bond. Look for the questions. A transistor has, next question, a transistor has one p-n junction, two p-n junction, four p-n junction, no p-n junction. That we already draw here. So this is the block diagram of a transistor and the circuit diagram of a transistor. There is a P, N, P. These are three terminals, and then P N forms one PN junction. Then this NP forms another PN junction. So this is a two PN junction transistor. That's actually say it's a bipolar PN junction transistor. Okay. Which among the following is a consequence of inelastic scattering of light? Which among the following is a consequence of inelastic scattering of light? What is inelastic scattering of light? When the scattering occurs, scattering is one of the phenomena is the light matter interaction. When the scattering of the light occurs, some of the energy of the light is transferred to the scattering material such that the light wavelength also have some change. So what is the effect here? You know that in the case of Raman effect, there is actually happening a change in wavelength. It's a stock shift as well as a anti-stock shift, which is corresponding to the change in wavelength of the light. So that is actually due to the inelastic scattering of the light. Okay. Lasing action requires which of the following phenomena? Stimulated emission, amplification of particular frequency emitted by the system, none of this, all of this. So we already have the full form of laser. What is the laser full form? It's light amplification by stimulated emission of radiation. So there's light amplification by stimulated emission of radiation. What's happening? Using stimulated emission of radiation, light is getting amplified. This phenomenon is known as a laser. So this technique is known as the laser. So what are happening there? Stimulated emission is happening. Due to stimulated emission, light amplification is also happening. So the amplification of light is happening and stimulated emission is also happening. So the answer is A is correct, B is correct. So the most appropriate answer of this question is all of this. A laser beam is used in surgical procedure because laser beam is in the case of laser we are actually using the cutting and other things. So why we are mainly using in the case of a surgical procedure, the thing is that the cut portion is will be very minimal. So if you're using at a light you can focus, and the focused beam will be like a micrometer size and the cut portion will have a micrometer dimensions, so that will be very good, that can cut in a precise manner, damaging very minimal number of tissues and other things. That's why laser is used in the surgical procedure, because it will be very sharp. So it will be sharply focused in the small area. Okay. So now the function of a sensor is to dash measure the speed of light, separate classify physical parameter, detect events within a specific environment. All of this. So just think that what can be a sensor. It has to sense something. It has to sense some physical phenomena, and either it can sense the whole physical phenomena. No, it can have a limitation. It can have select some region of the particular phenomena. For example, eye is a sensor, and what it is detecting, actually it is detecting light. Whether it can detect all the light, all the ultraviolet radiation. Light is actually one of the ultraviolet radiation, and whether it can detect all the ultraviolet radiation, no, it cannot detect all the ultraviolet radiation. It can only detect there's a wavelength region. That is what you are called our range of visibility, which will become around 400 nanometer to 700 nanometer, what we are calling as VGOR. And when this frequency, or the when this wavelength of light is actually falls in our eyes can create some signals, and that our brain can interpret and give some effect for that. So that's a sensor. What is called, it can detect a particular events or a specific environment. It can convert one form of energy to another form in a specific range. What's a metastable state? Where the metastable state is coming. Where the effect of metastable state is coming. You already know that the metastable state is coming in that lesser thing. So what is a metastable state? A very excited state is a metastable state? No. Because atoms can go if there's a corresponding particular wavelength or the corresponding energy, if atoms are obtained, it can, all atoms can go in an excited state. Whether that excited, all the excited state can be called as a metastable state. No. Says that have a shorter lifetime than ordinary excited state. So what means a shorter lifetime? It will immediately go there and come back. Then actually what is meant by that one? So the stability at that particular state is less. If it is coming back very fast, so it is having shorter lifetime means it has a less stability. So it is not metastable. A highly unstable state. Highly unstable state means it cannot even stay there. It is not at all stable. So that is also not a metastable state. A stable intermediate state of the system. So that's actually the metastable state. So what happens in the case of a material? And don't think that all materials have metastable state. Okay, there will be different material. Some material may have metastable state. Some material may not have. Depends on our application. We may have to find out something in a metastable state. For example, ruby have a tertiary metastable state. So atoms can go to its excited state, then have some change in energy, and it can go to metastable state. There, the lifetime will be more, such that the more and more electrons or the excited atoms can stay there. That what it will read is some photon with the same frequency happen here. This everything will go together, which is called the stimulated emission. One can trigger another can trigger another, such that it will be like a highly coherent unidirectional like this. Okay. So going to the next question. Optical fibers are used in MRI. MRI, what is the full form of MRI? MRI full form is magnetic resonance imaging. So what it's techniques which is using here. It's a nuclear magnetic resonance. So NMR is actually the techniques based on the MRI scan. So nuclear magnetic resonance is using, and based on that one it is doing an image. So here there is no light at all. So there is no optical fiber. Ultrasound scan, as the name suggested, it is a scanning method using ultrasonic waves. So this is actually sound waves which having a frequency which is beyond our audibility level. Our audibility level is a 20 hertz to 20 kilohertz, like whatever you told regarding C. Our seeing capabilities lies between 400 nanometer to 700 nanometer. So which have a frequency beyond 400 nanometer, that is called ultraviolet. So which have a frequency which is less than 700 nanometer, that's called infrared. Similar way, we have a audibility range also. All frequency sound we cannot hear. We can only hear the sound which is coming 20 hertz to 20,000 hertz, 20 hertz, 20 kilohertz. So if some frequency of the sound is beyond 20 kilohertz, that is called ultrasound. And some sound which have a frequency below 25 hertz, 20 hertz, is called infrasound. So these are mechanical waves. Sound waves are mechanical waves. So ultrasound, we cannot hear the ultrasound, but bats, dolphin, and other things can actually hear ultrasound. So we can use the ultrasound waves and use it as a method to image the tissues inside our systems, like the organs in our body and the fetus inside the womb. All the things can be imaged, and this technique is called ultrasonography or ultrasound scan. Here also sound waves are using, no light, non-optical tissue. PET scan. What is a PET scan? It's a positron emission tomographic scan. We are actually using some nuclear method only. So some positron emitting, that's some radioactive isotopes are actually we are using to understand the different conception of this can be radioactive particular materials which can use and will inject in our body, and we will see the consumption of that radioactive molecules along with the other molecules. So based on that one, we can do a scan that is called PET scan. That's a positron emission tomography. Actually, what we are detecting the emitted positrons. So the thing which we are undergone a medication of a nuclear medicine. Okay. Then it's the thing endoscopy. What's endoscopy? That's as the name suggests, it is like a looking inside, or scopy means something which we are imaging. Scope, microscope, telescope. So something we are looking. Endoscope. It's important. We are looking inside. How you look inside? There will be a bundle of fiber or optical fiber, which is called a light pipe. At one side of the light pipe, we will attach a light source, and we will put it inside our stomach by different means, such that the light bulb will give light to the inside part, and through this light which is coming through this optical fiber, we can visualize the inside portion of our body, and this method is called endoscopy. So only the light is using in endoscopy only. So since we are using it, it has to go through our stomach or some intestine area. It has to be flexible, and if you want to take light through some flexible thing, that should be an optical fiber. Otherwise, light is always travels in straight line. You cannot take a single medium to take light from a zigzag path. So there is definitely that optical fiber and the discovery side of the world. Okay. Going for the next question. Which of the following phenomena explains the wave nature of the light? It's Compton effect. How the Compton effect saying Compton effect is dealing with the light is interacting with the small particles like electrons, and it can change the direction of the hitting. Why the change in direction is happening? Because the light is like a light is interacted with the particle, and electrons also gain some kinetic energy, and due to this collision, some collision effect is happening. The case of Compton effect. So like usually what will collide? Particle will collide each other. So the Compton effect is actually giving the particle nature of the light. Okay. Photoelectric effect. Photoelectric effect is actually explained by the great scientist Albert Einstein. In that one, what it is saying, when a light having a particular energy or a particular frequency is coming and hitting on the metal surface, what happens? Electron is got ejected, and electron creates a photoelectric current. So the kinetic energy of the ejected electron is given by the some difference between the energy of the incident light and work function of the metal. So what's happening? It's like a law of conservation of energy. A high power or a high energy wavelength light is actually coming, and some of the energies are taken to eject the electron from the surface of the metal. That much energy is consumed. The remaining energy is actually converted to the kinetic energy of the electron. So this is also something like a photon is actually causing the emission of one electron. So this is like a, it's also saying something like a particle nature of the light. So each light is like a quantum of light. One quantum of light can eject one electron with a particular kinetic energy. Depends on the property in a wavelength of the light as well as the property of the material. We will discuss. Interference. What is interference? Interference is the phenomenon of when two lights coming together, or we can say that two coherent light sources are acting together at a position. What happens due to the superposition of the light? You can see some high intensity region as well as low intensity region. The low intensity region will be so prominent in such a way that that should be dark. So what happening is that, so if you are considering light as a wave, so the wave will have a crust and trough region. The crust and crust of the wave will interact and give a high intensity region, whereas the crust and trough region will interact and give a zero intensity region. So in the case of light, which is moving like a wave in a water body or something, it can produce a crust and trough, and the interaction of these particular waves can cause the dark also. So in the case of interference, we are using the wave nature of the light. Okay, so the answer is interference. എടോ നീ എടി ഇരിക്കുന്നേ? അങ്ങനെയാ നീ പോയിട്ട് അവിടെ ഹിമാലയ പോയിരുന്നോ? ആയി- ആയി- ആയിക്കോട്ടെ, ഞാൻ കൊതു പഠിക്കണ്ടടോ. ആ ആ ആ അന്ന ഒരു കാര്യം ചെയ്യോ, അടുത്ത ബസ് വരും, അന്നേരം കയറിയിട്ട് നേരെ പോയിട്ട് അപ്പുറത്തെ സ്റ്റോപ്പാണ് ഹിമാലയ. നീ ഇപ്പൊ ഗസ്റ്റ് ഹൗസിന്റെ നീ ഇപ്പൊ മുന്നോട്ട് നോക്കുമ്പോൾ കാണുന്ന ജിംഖാനിയാണോ? ആ, അവിടെ നിന്നോ, അത് അവിടെ വന്ന് നിൽക്കുന്ന ബസ്സിന്റെ അകത്ത് കയറിയിട്ട് അടുത്ത സ്റ്റോപ്പിൽ ഇറങ്ങിയാല് അത് ഹിമാലയ. ആ, ബെസ്റ്റ്. ഇനി ഒരു 10 മിനിറ്റ് അടുത്ത് വന്ന് നടന്നു. ഞാൻ അവിടെ കൊതുകടി കൊണ്ട് നിൽക്കണ്ട, അതുകൊണ്ടാന്ന് പറയും. ഞാൻ 8 മണിയാവും. അല്ലടോ, നീ ആടെ അത്രയും കൊതുകടി കൊണ്ടുണ്ടാന്ന് വെച്ചാൽ അവിടെ ആളുണ്ടാവില്ലേ, കടിക്കുന്നത് കുറയും. ആ ആ. ഞാൻ വിചാരിച്ചു നീ ആടെ പോയിട്ടാ നിൽക്കുന്നത്. ഞാൻ പക്ഷെ വേണമെങ്കിൽ നീ ഗസ്റ്റ് ഹൗസ്, ആ ആടെ കയറിയിട്ട് ആടെ ഇരിക്കേണ്ട സ്ഥലമുണ്ട്, നീ വേണമെങ്കിൽ അവിടെ പോയിരുന്നോ. ആ ഹാ 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 ആ ആ ആ. അലക്കുന്ന സോപ്പ് പൊടിയാ. ആ ആ, ഉണ്ട് ഉണ്ട്. അലക്കുന്ന സോപ്പ് വേണ്ടല്ലോ, സോപ്പ് പൊടി വരെ. ശരി എന്നാൽ. ഉം. നീ അതിൻ്റെ കൂടെ കയറിയിട്ട് പറഞ്ഞാൽ മതി, ബുക്ക് ചെയ്തിട്ടുണ്ട്. ആ, അതിന് ഞാൻ ബുക്ക് ചെയ്തതിന്റെ ഒരു ഇതുണ്ട്, എൻ്റെ ഒരു കെമിസ്ട്രി ആണ്, ഞാൻ വേണമെങ്കിൽ അത് ഫോട്ടോ എടുത്ത് അയച്ചു തരാം. ആ, ഇനി അവരത് പിന്നെ കയറാൻ വിടുന്നുണ്ടെങ്കിൽ ആ കയറിക്കോ. ആ, ഞാൻ വരുന്നു പറഞ്ഞു. Next question is the main function of cladding in optical fiber. You see that answers are to reduce optical interference, to protect the fiber, to guide the light in the core. All of this. Let's see. So which of sorry, the function of cladding of the optical fiber is let's see what's the main part of an optical fiber. Optical fiber is the device, you can say, device which works on the basis of a total internal reflection. What's the total internal reflection? There is a dense medium is happening when light is actually traveling from denser medium to rarer medium. What happens? It's going bend and away from the normal. So after a particular angle, what happens? It is going some particular angle. It will graze through the surface. It will graze through the surface, and that particular angle of incidence at which the angle of refraction is 90 degree is called the critical angle, or it can be said as theta c. So what happens if light is incidence at an angle which is more than critical angle? Light is incidenting at an angle is more than critical angle, then their light will reflect to the back to the denser medium, and that phenomenon is known as a total internal reflection, or TIR. Then how it can be useful in the case of optical fiber? What is an optical fiber? In the case of an optical fiber, there will be a core which is made up of there will be a core which is made up of glass or quartz, which is acting as a dense medium, and a core will be surrounded with a clad, which is comparatively rarer medium. What is dense and rarer means it's based on the refractive index we are saying. Okay, it is not based on any material density and other things. This is based on refractive index. Dense medium is a high refractive index, rarer means lower refractive index. What happens when a light wave is incidenting inside through this denser medium, and it will pass through this way. It is hitting. So this angle is more than critical angle. This will hit. So here also it will be critical and critical. Go like that. So all the light will pass through this one without much loss. And this phenomenon is known as a total tone reflection, and this thing is called a optical fiber. Okay, so using an optical fiber, the rarer medium is caught against denser medium in order to avoid this. So the cladding is provided in order to give the rarer medium such that dot and reflection is always ensured. Okay. Now the question is so the answer is to guide the light in the core. So it will cover the core as a rarer medium. So it will cover the core as a rarer medium, and it will ensure the total internal reflection, and it will ensure the path of the light through the core. So the following is the basic unit of heredity. So the following is the basic unit of heredity. RNA, proteins, genes, or lipids. So this is a direct question. So the basic unit of heredity is called the genes. So genes are the special portion of nucleic acid or DNA, which will be corresponding to a particular protein. So that will actually responsible for the expression of particular protein, and that protein is actually useful for the particular features of the organism. So the basic the, and these genes are actually transferred from one generation to the another generation. So the heredity's basic unit is actually genes. So the following are components of a nucleotide. So nucleotide is actually the nucleic acids, our long chain nucleic acid like in DNA or RNAs. Monomer is called a nucleotide. So the nucleotide contains a sugar molecule. There is a phosphate group and as a nitrogen base. So all three things are actually answer. So nitrogen, phosphate, and sugar. So the answer is so the answer is all of them. So two types of photoreceptors present in human eye are. So there are different types of photoreceptors are present in our eye. So they are called cone cells and rod cells. There are cone cells and rod cells. So rod cells are actually helps to see in limited amount of light, and that is actually used to see the black and white. So black and white, mm, what do you call, mm, perception of image is given by the rod cells, and they are rod shaped cells and containing a pigment called rhodopsin. And cone cells are actually responsible for the vision at bright light as well as the color vision, and they are in the conical shape. So, two types of photoreceptors in the human eye are rods and cones. Which of the following are cells of immune system? So we have platelets. Platelets are components of the blood, but that's actually helps for the clotting of the blood. Melanocytes. Melanocytes are the components which is responsible for the pigmentation on our skin. Osteocytes. Osteocytes are the cells which is present in bones. Monocytes also present in blood, but that is actually one of the white blood corpuscles, which is actually one of the part of our immune system. Which of these are examples of genetic vectors? Genetic vectors are bacteriophages, plasmids, virus. So bacteriophages can be something different, and there are like plasmids and viruses are there that will attack bacteria or the cell in the form of transforming the genetic material from its body to that particular cell, and that will attack, uh, and that will function as that particular cell's own system, that will such that it will get replicated and it will use consume all the resources, and like that just the cell will get destroyed. Similar way, these plasmids can actually get inside that particular cells, and if it is not like viruses, it cannot produce any particular organism after that. It's like viruses will multiply and then it will break the cell wall and come out. But in the case of plasmid, that will stay inside the cell, and that will give that bacteria some extra immune power and other things. So it will go along with the bacterial generations. So the answer is both B and C. Which of the following are used in the treatment of cancer? For example, radiation therapy. Radiation therapy is used in cancer. Chemotherapy. Chemotherapy is, yeah, radiation therapy is the method in which we destroy the cancerous cells using high energetic radio waves. Yeah, like cobalt 60, which is a strong emission emitter of gamma rays, and using these gamma rays, we can destroy the cancerous cells. Chemotherapy. Chemotherapy is the method through which we use chemical substances as medicines and try to kill the cancerous cells. Photodynamic therapy. Photodynamic therapy is the thing which will selectively use some medicine or bind to that particular area of the cancerous growth, and using that light waves we can destroy the cancerous cells. So all these things are correct. So the answer is all of this. So RT-PCR test. RT-PCR test is a reverse transcript polymerase chain reaction. The term nanotechnology was coined by Norio Panichi. It's a direct question. The size of an atom is around zero point 0.1 nanometer. The material with two of three dimensions in the nano range, and the third is large Two of the size are nano range, and third is large. How it will be represented? Just you can just see. Two is in nano, x and y is in nano range, and z is long. What is this? It's a wire. So let's see what is the answer. It's quantum wire. Nanorods are examples of one-dimensional nanomaterials, 2D confined. So two dimensions are actually confined, and one dimension has a length. Which of the following is not a consequence of enhanced surface to volume ratio in nanomaterials? Increasing surface energy is not a consequence of enhanced surface to volume ratio. Increasing surface energy, increase in internal cohesive energy, reduction melting point, change in energy level per state. So, change in energy level per state is a consequence of quantum confinement effect in nanomaterials, not because of the volume, surface to volume ratio. Quantum dots can be used for quantum mechanics, machine learning, crystallography, optoelectronics. So, this is like a direct question. So, the answer is optoelectronics. We can use it quantum dot based solar cells, quantum based light emitting diodes, and different types are there. It's actually quantum dots are it's like a zero dimension confined, not like very small entities. It's all the dimensions are in nanometer regime. It can be used for optoelectronics. Like high coherence of laser is helpful in the following application: polarization of the light. Polarization and coherence are two independent phenomena. Coherence is actually saying that how all the waves are exactly in phase. Polarization tells about how the transverse wave is vibrating in a plane in the perpendicular direction of the propagation. So reflection of the light, refraction of the light, reflection of the light. All the lights, irrespective of coherence, light will get reflected. Refraction also. Interference of the light. In order to happen interference, and it should interference will also happen in all the wavelength. So, but the interference effect to be visible, the light should be in the coherent band. Okay. So these are the questions which is covering the unit week zero assignment solutions. So just we'll go to the week one assignment solution. So in a single slit diffraction, a circular aperture produces a diffraction patterns of concentric rings that grow fainter as we move away from the center. These are known as the answer is the that's known as airy disk. So in the case of a single slit diffraction, just we will have an aperture and light is passing through this way. And you will see on the screen there will be a light, but on the geometric shadow region also you will see some ring kind of thing. Still get diminished. If you just take a cross section and plot the intensity, it will be like this way. And this phenomenon is called as a Ayres disk. and this is coming because of the diffraction of the light through this aperture, and there is a limit at which we can go the dimension smaller. So we are having a limitation on the dimension of this aperture. The image which we are getting as in the form of a ray disk is called the refraction detected image. So we cannot take any image which is better than that one, because the diffraction phenomena we cannot suppress. Now there are different methods like super resolution microscopy that try to minimize the diffraction limits. Okay. So going to the next question. Which techniques is used for trapping and manipulating small particles using focused laser beams? That we already discussed in the last 2 weeks before. That is called optical tweezer, laser tweezer and optical tracks. Techniques involves highly focused laser beams to create a spatial density gradient used to trap and manipulate small particles, such as cells, viruses, individual molecules. When we can trap a polystyrene beads, optical traps. Sunlight is the main component of the synthesis of vitamin that we already know. That's called vitamin D. It is called calciferol. Okay. Question 4. What is the advantage using the biophotonics for studying in cellular processes? Why we are using biophotonics? Higher cost. That's actually not an advantage. It's a disadvantage. If it is invasiveness, that's actually an advantage. It's invasiveness. No. Invasiveness means it will penetrate through the cells. So non-invasiveness is an advantage thing. Slower imaging speed, that is not a advantage. So non-invasiveness is an advantage thing. So question 5. Which of the biophotonic techniques is commonly used for studying the structure and function of molecules in biological samples? Nuclear magnetic resonance is not allowed by biophotonics techniques. So there is no photonics at all. Raman spectroscopy and mass spectrometry, there is no photons. Infrared spectrometry. So Raman spectrometry is the biophotonic techniques for studying the vibration of the molecules. Hello, is it audible? So the session is actually around to wind up. I'm actually discussing the previous assignment questions and assignment zero, which actually discussed, and now we are going through this assignment. What it's like a revision kind of things. If it's audible and you have any doubt and other things, actually you can give in the comment box. So I'm sharing the screen also. So we are going to the question 6. For fluorescence microscope, fluorophore DAPI used to stain. So these are the things which is actually discussed in the class. So we can see what are the what is actually fluorescence microscope. What will be there will be a fluorescing source and there will be a filter. So what happens when your samples will be have any particular fluorescent properties? Either it can be its own fluorescent property or it can be labeled with some particular fluorescing type. Then we will excite with a particular wave and that will use for the excitation of the fluorophore and it will emit the fluorescent light and we will use a particular filter in such a way that excitation light will not come to the filter, only the emitted light will come. So you will get a better contrast. So one of the dye or the fluorophore which is using in the fluorescence imaging is a DAPI. So this is actually a DNA specific stain. So that can be used. Cytoplasm will not have DNA, microtubules and neurons. So the mainly the nucleus will be get stained when you are using a DAPI stain. So, which of the biophotonics techniques is commonly used in non-invasive imaging or tissues and in living cell organisms? So, magnetic resonance imaging, which we already told, it is like a magnetic resonance imaging is a imaging techniques which is running based on the phenomena called nuclear magnetic resonance. So, MRI is not the answer. X-ray radiography. X-ray radiography is using X-rays to image the thing, whatever the bone and other things. So this is not a method. It's not a, and high contrast X-ray is not at all good for cells also. Positron emission tomography is actually a method which using the emitted positrons. So some nuclear emitting medicine or material will be infused inside the body. It can be infusion or it can be in the form of drugs. It is actually given to the body, and by monitoring the emission of the positron from the body, actually we can get an image, and we can say that the consumption of different materials in at the different part of our body. It's mainly used to detect the cancerous growth and propagation of the cancer and other things. But yeah, optical coherent tomography is the photonic based method usually used to image the retinas and other things. So, so for non-invasive imaging of tissues and cell, providing detailed and real-time information. It's actually one of the good methods. Is that, and now mainly actually optical coherent tomography is now used for detection of cells inside our eyes. So, question 8. Which property of a medium is responsible for the dispersion of waves considering different group velocities and different frequencies? So, absorption coefficient, no. Absorption coefficient is like mainly depends on all the things. Density and elasticity is not the one. So, refractive index have a different thing. Even absorption coefficient has a mm, contribution from the refractive index itself. So the refractive index will have different method, things like that, how this getting bent as well as dispersion happens, all the things. So it will have a real part and imaginary part. So and the dispersion is mainly depends on the refractive index. So key factor influence and dispersion of waves in a medium. Different refractive indices for different frequencies result in varying phase and group velocities. So when you are saying that when a light, a polychromatic light means a light containing a lot of wavelength is passing through a material. Each material will have a different refractive index depends on the wavelength of the light. For example, red will have less refractive index, while it will have very high refractive index. What is the thing? Red will travel faster compared to violet. So what happens? The velocity or the speed of the light inside the medium will be different. If the medium is in the form of a prism and other things, this change in velocities actually result in the splitting of the light. That is actually we call as a dispersion. So the question, what is the most common causative agent for urinary tract infection? These are actually direct answer. This is we discussed in the class. This is E. coli, and that's a bacteria species. How are photons and electrons are being similar? So as a quantum particle, we can say that photons sometimes before this quantum mechanics emergence, we thought that electron is like a particle and photon is something like a light. So that's energy or wave. Now, after the advancement of quantum mechanics, it is known that electron can act as both particle and wave nature. Similarly, photon can also act as both particle and wave nature. So, so that answer is C. Both can behave like a wave. So, this is the dual nature of matter and radiation. So hopefully this session is good for understanding and solving your problems. And if you have any doubts and any clarification required, you free feel to put it in the doubt clearing session or the Google groups, which is you are a part in that. So the TAs will go through that your doubts and try to answer that one. So if you have any doubt, you can actually also ask in the next session. I think that next session will be almost like a last session for this course, such that yeah, you are have to appear for the examination if you are registered for this certification program. And I wish all the best for all. Thank you. If you have any doubt, you can ask. So I'm closing the session and wishing you all the best for the exam. Thank you.